cell component, and innate is a really big chapter, right? It's like huge. And adaptive is enormous, that I even split it into two topics, adaptive and disorders. So yeah, you guys have Connect assignments for these, but at this point I don't really expect you guys to have worked on the innate one just yet until we finish the topic of innate. And so we're still on innate, right? And we will be a little bit longer. So not so worried about that, but of course work on that discussion board, right, of immunology web pages. I haven't assigned a date to that yet. But you should have already read the first initial web pages, right? Yeah. Here, I'll click on it. Right, so when you guys go to our page even, it should have listed off to the side, right, in your to-do list. For me, it's stuff that I've got to grade, right? Oh. Yeah, I didn't think I had put a due date yet. No, I don't have a due date for that one. I had a previous already due date for the web pages, right? So I know we have more than 22 people in this class, right? So there's only 22 for me to grade. So that means about 10 people maybe have missed this assignment in the module. So make sure you get that done, right? So immunology web pages. I had already turned on the discussion board, and some people have started working on it. I just didn't put it in the unit. So I'll put it in the unit. But I haven't assigned a due date to that yet. But, start working on it if you want to, right? Get ahead of the game. I don't usually assign it until we finish innate. But I know some of you guys like to work ahead. We have one test left that you're going to take in the testing center. The other is take-home test. Remember, right here, you can take this at any time. You got to finish it by May 2nd. It's on all the case studies. None of this is on the final. None of the case study stuff is on the final, right? And before you do that test, make sure you do the Listeria assignment first, right? And read through, right? Watch the videos for all of these case studies. Maybe even print out your notes. Maybe even print out the PowerPoints that I have, right? Go through them all, get it all organized, and then open this test, because you can only open that test once, right? That's it. Right. Got it done. One thing off the list. Yeah, get it done. Get her done. Okay. All right. So, yeah, we just have this last unit left, which is huge. It's going to take us the rest of the semester to cover. Or let me put it this way. This month, y'all, this is it. Right? Which I hope, and I don't think this is the case for anybody in this class, but today's drop day, right? So if you're wigged out in one of your other classes and you need to drop and you're forgetting about it, make sure you get it done today. Right? And teachers can't drop anymore, right? It's 100% students. So that's the bad thing about that, though, is people who've truly wigged out and stopped coming to my class, I can't boot them. They have to do it themselves or pay the consequences. All right, so last time we were talking about cells and those web pages I just referred to, right? I even took pictures from them, right, and put them in my PowerPoint. So I think we did a pretty good job of going over the cells last time, and even did a little poll everywhere quiz. We started talking about cell communication, right? So remember, these are the different groupings, like chemokines you need to understand in general. These are chemical signals, right? Basically the hormones of the immune system that are used for chemotaxis. The colony stimulating factors tell the stem cells in the bone marrow which white blood cells we need. Interleukins, there's a lot of these, right? They talk in between white blood cells to each other. Interferons, remember, always think viral, and we're going to talk about specifically how interferon works a little bit later. So those are the general classes, right? We don't need to know these specific examples under here. But these are just to give you some ideas of how these things work. And then we've already seen adhesion molecules in action, right? Right at the very beginning when we looked at inflammation, right? They're really important for getting those white blood cells to that area, allowing them to be able to adhere. So communication is key, right, for our immune system. And so to be able to communicate, we have specialized receptors on our cells, within our cells, that help us recognize when invasion has happened, right? When the bad guys have gotten in. The question we're asking here is, what's the outcome? So what happens if a toll-like receptor is engaged, right? What happens? When complement system is activated, what happens? Yeah, we'll talk about it. Right? And what happens when we detect double-stranded RNA? Remember, double-stranded RNA comes from what type of pathogens? Do we have double-stranded RNA in our cells?
No, that's viruses.
That's viruses, right? That's viruses. So that, again, that's aimed towards viruses. So we have sensory systems within the blood that detect signs of tissue damage or microbial invasion. And the important thing is they respond to patterns associated with danger. And some of this will cause the direct destruction of the microbe, right? Some of these systems, when they become activated, we will directly destroy the microbe. Or in addition, we'll recruit cells and other host defenses to the area to eliminate the invader. And if we can't at least eliminate them, we're going to trap them in, right? We're going to trap them in as best we can. So what are some common patterns, molecular patterns, right? Common molecules associated with bacteria. So we can even divide bacteria into our two major groups, gram-positive and gram-negative. Remember, gram-positive are positive because of what cellular component? The peptidoglycan, right? That very thick cell wall layer made out of the molecular component we know as peptidoglycan. So that's very common as it relates to especially gram-positive bacteria, right? So wouldn't it make sense evolutionary-wise that we'd have a receptor specifically for peptidoglycan? Sure, right? We recognize that that is associated with something bad, right? Even though we have bacteria, remember, that live in our intestines and may live on our skin, we've developed a symbiotic relationship with bacteria being there. It's when bacteria get into our tissues, right? Under the skin, into the tissue, right? Out of the intestines, maybe into the abdominal cavity, right? We certainly don't want them there. So we have a way of detecting their presence there, right? Where they're not supposed to be. So peptidoglycan is one, right? Toll-like receptor, right, that we can detect that bacteria is where it's not supposed to be, especially gram-positives. Now, what about our gram-negatives? What outer membrane component do gram-negatives have? It's abbreviated LPS. Lipo. You got it, Grace. Polysaccharides, right? Lipopolysaccharides. So there's a lipid component and many sugars, poly meaning many sugars associated with it. The flagella protein, made up of the flagella for bacteria, that particular protein, we have a receptor for. So, again, they use a lot of acronyms here, right? But the important thing is right, we're recognizing patterns, molecular patterns, right down to the molecular level, that are usually associated with the bad guys, right? So sometimes referred to as microbe, but sometimes even referred to as danger. I don't want you guys focusing on that. I want you guys to think about it even in more detail as we just talked about. Think about the molecules themselves, right? What are the common molecules that say bacteria, right? That say virus? There are things like lipopolysaccharides and peptidoglycan and lipotectonic acid and lipoproteins and flagellin subunits and viral RNA molecules. We can tell the difference not only from our own RNA, but we can tell the difference between viral RNA even if it's not double-stranded, because we process our RNA differently, right? So we are able to detect if it's not ours. So we're going to play a short video Let me make sure I got my audio plugged in. To help us visualize this.
The surfaces of phagocytes, called pattern recognition receptors, make it possible for the phagocyte to immediately recognize a number of different components of infectious agents, referred to as pathogen-associated microbial patterns. Toll-like receptors are a class of pattern recognition receptors that play a critical role in the early innate immune response to invading pathogens. Several different toll-like receptors located in the cell membrane or endosomal membranes of phagocytic cells have been identified. They recognize lipoproteins and lipoteichoic acids, viral double-stranded RNA, lipopolysaccharide, and bacterial flagellum. Some toll-like receptors exist as monomers, whereas others...
All right, they're going to start getting detailed. One thing I want to point out here, right? Notice this receptor. What did they say this is for? So this is toll-like receptor 3. So there's toll-like receptor 3. It's for viral double-stranded RNA. Why isn't this receptor on the membrane? Why is it inside the cell?
In case the virus gets in.
Because the virus is going to get in, right? That's where viruses have to go, is inside of our cells. Now, this is a phagocytic cell, so it's going to engulf it anyways, right? But if it was to get into this cell, you want that type of detection on the inside, because the RNA doesn't become out of its protein coat until it gets inside of our cells, right? So it makes sense that that type of protection is on the inside. And we have other receptors, too, within the cytoplasm, within endosomes, right? Again, for internal invasion. We can detect peptidoglycan on the outside, but we also can detect it on the inside. Now, for a phagocytic cell, why is it important that they have so many different toll-like receptors? What does a phagocyte do? It engulfs. Now, how does it engulf? The very first step, it must what?
Have an antibody?
Well, if it's not coated in antibodies, right? How is that phagocytic cell going to know to eat that bacteria? It has to recognize. So how is it recognizing? These toll-like receptors, right? So if it's not coated in C3b, which we're going to talk about in a moment, that's a product of complement, it's not coated in antibodies, we have to have some way of recognizing, right? And physically attaching to it, right? This is going to be an additional added benefit of it's going to allow that phagocytic cell to grab onto it, right, and engulf it. And then, of course, phagocytosis means that it's going to do what with it once it gets inside? It's going to destroy it, right? It's going to destroy it. It's going to use its lysosomes, those digestive enzymes in those vacuoles to destroy it. So who are our phagocytic cells? Who are our main phagocytic cells? Who's already in your tissues waiting for the bad guys to breach the barrier? Macrophages. And the other guy. One more. Starts with a D. Dendritic cells. And who are we going to call in think about it, that macrophage, right? So think about this. So the macrophage detects, say, lipopolysaccharide and engulfs a gram-negative bacteria. Is that it? Is that cell going to go, ooh, yum, snack? So these things play two roles, right? Because now this is a receptor, which means that it's receiving information, right? So where is that signal going to be sent for this cell? We won't go through all their little tiny molecular mumbo jumbo, right? I'm going to fast forward over this. Right? So they're going very detailed, right, at the molecular level, the different molecules that are involved. But we're basically going to send this message, whoo, look where it's going, into the nucleus. Because remember, that's the command center, right? So now a signal is being sent at the nucleus, and the cell is going to do something, right? It's going to respond. So in this case, if you've got a cut in your skin, and bacteria get in, gram-negative bacteria get in, phagocytic cell, whether it be a macrophage or a dendritic cell, engulfs it, destroys it, but also we've signaled this cell, we've confirmed that this is a bad guy. So now he is going to utilize information in his DNA to make a signal, right? That's going to probably tell the surrounding cells what? Something's not right in Denmark, right? So if we've got invasion, what do we want to happen next? What do we want that phagocytic cell to tell the cells near it? Activate. What's our most common response for invasion? It's our most common immunological response. Who do we want to come and how are they going to get there? We want white blood cells. More specifically, we want more eaters. Who's our big, powerful eaters that we're going to call? Because they're not on site. The macrophages are there. The dendritic cells are there, right? We have resident cells. They're going to call what reinforcements in? Neutrophils. And that process of calling them in and getting them to that area is generally referred to as, you're going to start swelling. It's going to get red and hot. It's called inflammation. Right? And this all starts with, right, triggering these receptors and this cell sending out those signals. That's the outcome here, right? Cytokines are released, right? Chemical messages. Neutrophil attractant saying, hey, come here, I need you. Or maybe they'll call in B cells. Or maybe they'll call in T cells. Or maybe you need tumor necrosis factor. Depends on which receptor was triggered, right, as to what type of response we're going to elicit. So that information that's transmitted all the way to the nucleus where gene expression happens, right, where we're going to express the genetic information that's going to allow them to send out signals, right? Maybe it's an inflammatory response. Maybe it's an antiviral response. Those are our two most common outcomes, right? If we've got invasion, we want to respond. If it's bacterial, it's probably going to be an inflammatory response. We want those neutrophils there eating up the bad guys. If it's a virus, we want that type of response. So that's why, each one of these receptors allow us to detect different common elements, but we're also going to respond in a particular way, depending on that signal. So macrophages and dendritic cells definitely have these. It's going to help them aid in grabbing on and phagocytosing, but also signaling the system as to what is invaded. But other cells in our body have toll-like receptors as well, places where bacteria are not supposed to be, so that we can signal when invasion has happened, so we can sense, we can send out the alarm. Now, phagocytes and dendritic cells are great because not only can they recognize, they can also kill, right? Not all of our cells can do that, right? So they definitely need to be able to signal for help. So NOD-like receptors are found in our cytoplasm, and again, very similar molecular patterns, right, that's associated with danger and pathogens. Flagellin protein, peptidoglycan, double-stranded RNA, single-stranded RNA that doesn't look like our RNA because it's not capped. We cap our RNA. So a lot of times, once these are triggered, these cytoplasmic proteins will form what's referred to as an inflammasome. So what type of response do you think that that's going to trigger? An inflammatory response, hence why it's called that, right? That one's a pretty easy one. It's a dead giveaway. But again, what's inflammation? It's getting the white blood cells, especially our neutrophils, there, right, to destroy the invaders. So that's it for our cells lecture. We're going to move on now to Part C, which is our last one for innate, which is first line of defense. So as I said, I kind of jumped ahead, right? Especially with your current textbook. And really kind of started with the inflammatory response because it's so important, and looked at the cells involved. And now we're going to kind of back up and talk about, right, what's our very first line of defense, and do kind of a general overview of innate. So our first line defenses are our anatomical barriers, right? So these are literally the walls that keep the bad guys out. So what's our wall? It mostly is going to keep the bacteria and the viruses out.
Skin.
Our skin. But we do have openings in our skin, right? A digestive tract is a tube that runs straight through our body. But this is basically a big opening too, right? What do we have that protects that entire digestive tract?
The mouth and the nose.
No, it's a sticky substance. Mucus, right? For our eyes, it's a specialized mucus. It's referred to as, for our eyes, what do our eyes produce? Tears, right? Our mouth, saliva, right? Still has mucus associated with it, but it also has digestive enzymes and other things that helps with the breaking down food, but it also contains things that are antimicrobial as well, right? Your tears do, right? And again, your tears are to bathe and wash your eyes continually. You're always producing it. Only when you excess produce it, right, does it run down your face in what we refer to as tears, right? But it's always being produced, and it goes down your nasolabial duct, right here, next to your nose, right into your nose, your actual nose on the inside, your nasal cavity, which then, of course, is connected to your throat, right, which is connected to your digestive tract. So there's one other system, so your eyes, your nose, right, your mouth, all of this connected to the throat, okay, which goes down the digestive tract and then out the body. Right, so we can wash everything on its way. Well, we run into a problem, though, when we get to respiratory, though, right?
Yeah.
Because this is an ended system, right? So stuff goes in, how do we get it out? We can still trap it in mucus, but we need an additional system to be able to get the mucus out. Anyone know what that is? Coughing helps, but you're actually continually always doing this. You have ciliated cells associated with your respiratory tract, which beat and move the mucus up to guess where? Your throat again. So you can swallow it and wash it out the system. Right? For your reproductive tract, especially for women, again, bathed in mucus. Right? Urinary tract. What protects you with that is just literally going pee. The flushing action of cleaning out the bladder helps stop stuff from going up the bladder, up the ureters to your kidneys, right, which is really bad news, right? So drink lots of water, pee often, keep flushing out the system. Don't let them stay long enough to travel north. So we just talked about our pattern recognition receptors, right? These are part of our sensor system. Another component of our system that we're going to look at is complement. And pattern recognition receptor interferon response, which is our viral response, really falls under, again, that pattern recognition, using receptors to detect viruses. But again, there's a specialized response for viruses as opposed to bacterial infection, which usually is going to be an inflammatory response. We've talked about our phagocytes, right? Cytokines, remember these are the chemical messages. These are how the communication happens. We've already looked at inflammation. And lastly, we're going to talk about fever. So this was meant for if we had already reviewed, right? But this is good. We'll review right after we went over who was paying attention today, right? So what are the common bacterial molecules that toll-like receptors recognize? right? Or we could say which of the following are not bacterial molecules, should be plural, recognized by toll-like receptors. Which one of these is not bacterial? Double-stranded RNA. What does that say? Viruses. One brave soul. Which white blood cells phagocytize invaders, dead cells, and debris? All of them. Remember, monocytes are what your macrophages are called in your blood. Macrophages, right? That was a tricky one. Tricky now, so you won't hate me on the exam day. Okay, which class of cytokines tells the bone marrow hemopoietic stem cells to make more white blood cells? Yes, the colony stimulating factors. And again, think of white blood cells that are all the same as a colony, right? As a group. That one usually helps. This one, I couldn't really ask this one because there's multiple correct answers. Which class of cytokines do white blood cells use to communicate with each other? Pretty much all of these, right? But I was really looking for interleukins here, right? But pretty much these are all used by white blood cells. So I had to make it a more specific question. This is when you write new questions and you realize, you test them out and you realize they're not good. It's too general. The good news is the ones on my test are tried and true. All of the following trigger inflammatory response except. So engagement of those pattern recognition receptors, do you think that could trigger inflammation? Right? If we see peptidoglycan, are we going to trigger? Probably. Complement activation? Yes. Yeah. And we're going to learn about that today. Interferon induction of antiviral protein synthesis. This is the one that's not going to trigger inflammation. And of course, we already know tissue damage will trigger inflammation. The reason why I put this one up there, y'all, guess where I got this question? Right out of the book. Right out of the book. Right? Don't forget, right? You guys, lots of resources literally right at your fingertips. So many times students forget, right? Those are good things to read over at the end of the chapter. See if you can answer those questions. Who knows? I might steal one and put it on the test. All right, so again, it's a security system, right? Our immune system is a security system in that it's going to have these walls, right? Our skin, our mucus, and within that we have antimicrobial substances. We have sensory systems, right? They're going to help us detect. One of them is those pattern recognition receptors we've already talked about. The other is complement. They're going to talk about in a moment. And then we want to, we don't want to let them in, right? So we're going to have these barriers, but they're going to get in, right? So we have to have a way to sense that they got in. But then just that is not enough, right? We don't want to be like, oh, the bad guy got in. So what, right? Okay. We want to be able to eliminate them, right? And so we want phagocytic cells, right? We want interferon response for viral attack. We want the activation of the complement system, which can lead to cell lysis and cell death. We want inflammatory response. We want white blood cells to get to the area if we've got invasion, especially with bacteria. We want to turn on our fever response if it's going to be helpful. And all of these are meant to help eliminate the invader. But again, this is very generalized, right? We're just talking about bacteria in general. We're talking about viruses in general, right? We're not getting very specific here. So what are our first line defenses, right? Including our physical barriers antimicrobial substances, and what role do the normal microbiota play? So our skin itself, right, we have two main layers. We have the epidermis, right, and that's where transient bacteria, flora would be, right? They're just catching a free ride, right? The ones that actually live in our skin are living within the dermis layer, the deeper layer that generates the epidermis layer. One of the areas for concern for us, too, that can also get infected is the hair follicle or the glands associated with the follicle. These are sometimes just commonly referred to as oil glands, but the proper name is your sebaceous glands. And, of course, they produce an oily substance. The reason for that is so that the hair will slide out of the hair shaft, right, out of the hair follicle. Where mammals were covered in head to toe with hair, right? The unfortunate thing, there are some microbes that like to eat the sebum, right, this oily substance. And for some individuals, you overproduce this, so much so that you create a really good environment for the overgrowth of the microorganisms. So when you commonly get this type of infection, what do we call this? Zits, acne, pimples, right? And so, again, it's normal bacteria that most of us are exposed with, might already have on our skin. The problem a lot of times is that they're overgrowing, right, or getting trapped inside. And I know the very first indication we always want to do is to pop that, right? Right? And so what are you doing? You're releasing infection to the surface of the skin now. So then that can spread to other nearby hair follicles. And when you're doing that, you're creating tissue damage. So there is the potential, too, that it goes out of the hair shaft and out of the sebaceous gland into the surrounding dermis. So you could even make the situation worse. Although it's really hard to resist that urge, right? The best course of action, it's a bacterial infection, right? A little bit of triple antibiotic ointment, good old Neosporin. Most of us have it in our medicine cabinet. Just put a little bit on it before you go to bed at night, a couple nights, it's usually be gone. Now, some people, of course, have more than one or two zits, though, right, that have a condition referred to as acne. And again, that's usually an underlying condition of the fact that you're producing too much oil, right? You're giving them too much food, right? So there are other treatments for that, right? And part of it, too, you know, for some people, you know, that thing they're always, proactive, right? It makes sense, you know, if you clean, right, that there's potential that could be helpful, too. It just depends on what the underpinning reason for that constant infection is. Make sense? Right. So unfortunately, as I've said numerous times, right, we're a big fat juicy cheeseburgers bacteria, right? They love to eat us, right? We are yummy, yummy, right? And if we provide them with the habitat and the food, they're going to eat it and they're going to grow, right? And sometimes to quite large numbers, so much so that you have a visible infection of the hair follicle. So it is a barrier, though, right? What's beneficial about our skin? What about that very outer layer?
You wash it off?
You can wash it off, right? You're continually losing, right? Those are dead cells. You're continually sloughing off your skin layer. You're continually replacing it, right? As you're doing that, anything that lands that doesn't take up residency, that's transient, gets pushed off as well. 90% of the dust in your home, for the most part, for most people, is your epithelial cells, right? And then that's what the dust mites eat, is your epithelial cells, right? Of course, there's a large dirt component too, depending on who lives in your house. If you have kids and animals, right, it could be largely dirt and not so much, but it still would be. So what are the aspects of skin that help prevent infection? The dermis layer is very tightly woven fibrous connective tissue. So that's really difficult for microbes to penetrate, right? Unless we injure it, right? Unless we get a cut, unless we can't resist and we pop that zit. The epidermis is many layers epithelial cells, and the outermost layers we said is dead. And it's actually filled with keratin, which repels water, which for the most part maintains a relatively dry environment, which isn't conducive to microbial growth, right? They need water just like we do. So if there isn't a lot of water, they're not going to grow. And as we said, we're continually sloughing this layer off. So this is an interesting diagram from your book, but it's pretty good. Right? It's very basic, right? But our mucous membranes, right, protect all the openings, right? So that whole digestive tract and respiratory tract, as I said too, which is actually linked to your digestive tract. We constantly bathe all of this in mucus. It's a gelatinous, sticky substance, and that's exactly what it's meant to do, is to have the bacteria, the viruses and stuff get stuck in it, right? So that then we push it along where we want it to go, right? So how are the microbes trapped in the mucus and removed? So for our digestive system, right, we're trapping in the mucus, and the normal muscular contractions we refer to as peristalsis just move stuff through the system. So this is another problem too. If you haven't pooped in 3 days, whew, that's not good, right? You got to get that stuff out. It needs to leave. Because the longer it stays, right, the chances are it can move from the gut into the body. And we don't want that. For our respiratory system, as I said, it's a little bit more complicated. Other than just the mucus, we have the mucociliary escalator, is referred to as. It's this system of the mucus along with the ciliated cells, which actually beat upward, right, to move the mucus up out of the lungs into the throat and then pass it through the digestive system. So this is continually happening. You're continually breathing in crud, right? You're trapping in the mucus in your respiratory tract, and you're moving it up and out. This, of course, we recognize happens more so with infection, right? You get increased mucus production. So much so, you may even have stimulation of the cough reflex, right? Because of the excess mucus moving through the system. So we also produce within our mucous membranes and even in our skin antimicrobial substances. So salt, which I think the reason why we produce salt isn't actually in our sweat, isn't actually to be antimicrobial. I think it's one of those side benefits we have, right? But the one good thing is because our skin will tend to get salty, especially more so when we sweat, non-salt tolerant organisms can't grow. But of course, the ones that we've developed a symbiotic relationship with, like Staphylococci, epididymis, it's salt tolerant, right? That's why it can grow on our skin, because it tolerates salt, right? And so one of the plate media, right, that we'll look at and I'll have out for us at the expo too, is the mannitol salt agar. We use this to grow Staphylococci because, again, they're tolerant to high concentrations of salt. So the fact that there's a lot of salt in that media really helps us select for that group, especially when we're looking at clinical specimens. But anyone make homemade ice cream, the homemade ice cream machines? Right? What do you add to the, you add salt to the ice, right? And it kind of melts the ice a little bit, and it actually creates greater contact between the ice and and I don't understand the true physics behind it, I'm not a physics teacher. But there's something involved in that, right? We're adding that salt. And I know from living in the Northeast, right, if you add salt to ice, it melts it, right? So you're melting it a little bit so that you have greater contact. I can tell you that in microbiology lab, when we do certain preparations where we need to put stuff on ice, we usually use an ice bath. We're using ice and water, right? That's colder than just ice itself because the water will get really cold because of the ice, right? And then it comes in contact with more of the surface area of the tube where we're doing our reactions in, right? Just ice by itself, right? You just don't get enough contact between it and the surface. Right? Can you imagine if we added salt to it? Right? It would be really cold. But as I said, we have this side benefit of, this fact that we produce the salt. So the whole point that I was trying to get to is, why is our sweat salty? So based on those situations I was just talking about, why do you think we produce the salt in our sweat? Why do we produce sweat? To cool, to create that convection to cool. And so apparently salt aids in that convection ability, that cooling ability that happens when we sweat. Kind of cool. So lysozyme, this is an enzyme that degrades peptidoglycan. So again, very beneficial for us to produce it in our mucus, and even in our modified mucus like our saliva and our tears. Peroxidase enzymes, these produce oxygen radicals, right? So these are very damaging forms of oxygen. So inside our phagocytic cells, we can produce this and help us destroy pathogens that they've engulfed in the phagosome. Lactoferrin, as the name implies, lactose, so you produce this, it's in your secretions, right? It binds iron. Why do we want to bind to iron? Well, we have iron in our red blood cells to help us carry oxygen, right? So we need it for, you could say, our metabolism. Do other organisms need iron? Yeah. Yeah. Bacteria need iron for their metabolism as well. So again, we're a rich source of iron, and especially our red blood cells, right, are an extremely rich source of iron. This is why some bacteria have adapted the ability to produce enzymes that will actually poke holes in our red blood cells, right? So I'll have Friday too, those blood agar plates, and those guys in lab, you're going to be inoculating these plates and stuff this week, or next week, depending on which lab you're in. And you will see if your organism produces these enzymes. Now, there's two different types. There's alpha and beta, right? How do we tell the difference between the two? Remember, I always say beta is bad. Why? What does it do to the red blood cells? It just completely destroys them, right? It'll completely destroy them. Alpha, on the other hand, just pokes holes in the red blood cells. It's incomplete lysis. So you get that greenish discolorization to the plate. So iron is very important, right? Think about those blood agar plates. Think about bacteria actually making specific enzymes to lyse red blood cells to get at what? Iron. That's the key. So again, do we want iron to be readily available to them? No. So we produce a carrier protein, lactoferrin, to bind to that iron and keep it away from the bacteria. Right? So then, if they have to or they can, they'll be more aggressive about it, right? They're going to go, okay, fine, I'm going to attack your red blood cells next. Right? Which is why strep throat hurts so darn much. Right? That one produces beta hemolysins. It completely destroys your red blood cells. It is one evil bacteria. He's serious in his assault. So you have transferrin in your blood. So again, what is it for? What's ferrin refer to? Iron. Trans, in this case, it's to help us transport iron in our blood, because iron isn't a very soluble substance. And remember, your blood is mostly what? Water, right? And so if it's not soluble, it's not going to travel well. Right? So transferrin helps it makes it more soluble, but also the added benefit of, again, where our proteins are binding to it, and hopefully we're going to keep it away from the bacteria. Defensins, these are amino acids, chains of amino acids that literally can insert into microbial membranes and create pores in them. We're basically throwing little daggers at them, right, and poking holes in them. So all of these substances, right, will be found in our mucus or in our phagocytic cells, or our modified mucus, our tears, and our saliva. Even in breast milk, right, they'll produce lactoferrin. So which body fluids do you think contain lysozyme? So this is an enzyme that does what? Digests and destroys what? Peptidoglycan. So would it be beneficial to be in our tears? Did bacteria get into our eyes?
Yes.
All the time. You're sitting here, y'all, you're getting bombarded. I guarantee you. Right? So would you want lysozyme there? You betcha. Are you ingesting? Are you taking into your mouth things that are contaminated with bacteria? Absolutely, all the time, even when you're breathing. Where's mucus? Bathing all these openings, right? So the digestive tract, the reproductive tract, the respiratory tract. Do you want lysozyme in that to help digest? Sure. What if they get into our tissues? The fluids that bathe our tissues, would we be beneficial to have lysozyme there? Yeah. In our blood? Yeah, so the answer is all of the above. Right? It makes sense, right, to have this ability everywhere we could possibly put it. This is probably the most abundantly produced enzyme by our bodies. So there are other components other than lysozyme and peroxidase and lactoferrin that help protect us. Our stomach itself has a very low pH. We actually make hydrochloric acid, which is a very powerful acid. And the reason for this is to aid in protein digestion, right? To be able to digest proteins. But if you swallow bacteria, right, or bacteria got into your eyes and got rinsed into your nose or came in your nose or your mouth, it's all going down the throat, right? Even if you inhaled them all the way into your lungs, they're going to get pushed, hopefully, back up maybe if they got trapped in the mucus and end up back down your throat. Everything goes into the esophagus and lands in the stomach, right? The stomach has an extremely thick layer of mucus to protect itself from invasion, but also from digesting itself, right? That very thick layer of mucus protects you from the acid and the protein digesting enzymes like pepsinogen you produce. If a bacteria lands in the stomach, what's going to happen to it? Hopefully it's going to get digested right along with the food that you eat, right? It's when we run into problems, when you run into pathogens that can resist this level, or you've ingested just so many, right? So think about it. If you ingest 10,000 salmonella bacteria, are all of them going to get killed in your stomach? Probably not. Some of them are maybe going to escape to the small intestines. And that's when we run into problems, right? As that they can infect the intestines, right? In the large intestine, you have normal microbiota. You already have E. coli there, one that's non-pathogenic that we developed a great relationship with, and a bunch of other ones that you hear about nowadays that they want you to take in probiotics, right? This microbial environment, what you digest in your stomach and your small intestines, you absorb in your small intestines. Anything you do not absorb passes into the large intestine. You do not do any digestion in the large intestine. You're basically absorbing water, last bit of vitamins or stuff like that, and vitamins that the bacteria now are going to produce because they're going to eat what you didn't eat. Right? This is the symbiotic relationship we have with them. They get to eat what we didn't eat and have a place to live. The good news is most of these guys produce vitamins that are necessary for us as well. As long as they stay in the intestines or leave with the feces, right, and don't travel out of the large intestines, and we don't have any problem with them. Right? We've developed this symbiotic. They're truly outside of our body, though, even though they're inside, right? It's a tube that goes through our body. It's actually considered an external environment. So one of the things that can happen, too, is that disease can be caused by a disruption of this environment, this little ecosystem we've got going on. So there are times when we take antibiotics, right? When you take antibiotics, does it specifically only kill salmonella? No. So say you have a salmonella infection. They give you antibiotics. It's going to kill off depending on what that antibiotic triggers. Like if it's all gram negatives, then guess what? Your poor E. coli's in the gut too are going to get targeted, right? And anybody else who's gram negative. If they give you a broad spectrum antibiotic that covers gram positive and gram negative, you're in trouble, right? They're gonna bombard all your poor babies in your large intestines. And if that population of bacteria, your normal flora, gets significantly reduced, that actually predisposes you to secondary infection with pathogens. Because when a pathogen comes to the system, part of the problem too is, right, got to make it past the stomach, right? And when it gets to the large intestine, if it gets there, it's got to compete, right? E. coli's already there. Other bacteria are already there. They're already taking up all the spots and eating all the food. And they want that spot, right? They're even willing to fight for it. Some of them even produce substances that are antimicrobial to other microbes. Because E. coli is like, this is my home. You can't have it. So usually, right, having those guys there, it helps outcompete them, right? Our normal flora help us out by being there. So the bad guys have nothing to eat and no place to land. They get pushed out the system. But you get a condition referred to as antibiotic-associated colitis, and the most common offender bacteria that will cause this infection of the intestines is Clostridium difficile. Because Clostridium difficile is a spore former found in the environment. So you're exposed to it all the time. But it goes right through your intestines, right? Because most of the time, all the other bacteria are like, no, 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 mine, 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 you can't have it, goodbye. But if they're gone and Clostridium difficile gets in there, then he's got free reign. The problem is he's a really evil pathogen. He produces toxins. He literally will eat your intestines. He is not a symbiotic guy, right? He's all about destroying you. And anyone who's worked in the healthcare field, do I got anybody just yet? Just wait, because from what I've heard, I haven't experienced it myself, thank goodness. But then again, if you've worked in microlab, we smell some really smelly stuff. But apparently, this one is an anaerobe, right, and produces gases. And I've been told that you know before you even get to that patient's room that they have this infection. It smells that bad. It has a distinctive odor, so I've got a couple of head bobs, right? You guys have experienced it. And one of the treatments will usually grosses people out, too. They'll actually do a fecal transplant, where they're going to, hopefully someone in your same household, because you'll have the same bacteria, they're going to take their poop and put it in your intestines to reestablish that microbial environment. Right? Because again, it's all about that balance. Right? Maintaining that balance. Okay, so as I said, with the bladder, the most important thing, how do we protect ourselves? Pee, 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 pee. Yep, lots of pee. Okay, for the vagina, again, this is a very important, delicate balance, right? The bacteria that are normally there, they produce a relatively acidic environment. This impedes other microbes from growing. But when you get a disruption, whether it becomes antibiotics or hormones or diet or any number of things or disease state, it can disrupt that normal balance that we have going on in the vagina and you get an infection. And one of the most common ones is a yeast infection. And the thing is, it's not really an infection. These are yeast you have already associated with your skin and for women, their vagina. But they overgrow if their bacteria aren't keeping them in check anymore, right? So if you knock down the bacteria with, say, antibiotic treatment, then the yeast take over. They overgrow because they're getting most of the food because they're no longer competing with the bacteria. You killed them off with the antibiotics, right? So that's another one, right, where you've got to reestablish, right? You got to knock down the yeast so that the bacteria can reestablish the environmental balance, right, that keeps everything happy. And if you're a woman who's never experienced a yeast infection, the rest of us are very jealous. Right? It happens to us all at one time or another. And it can happen to the skin, right? So really obese people too. The problem with the vagina is moisture too, right? Moist and warm all the time, right? And so skin too also has these normal yeast associated with it. When you're obese and you have the skin folds, you know what I'm talking about? And so then you have that moist, warm environment that can establish there where it's being trapped and it's not drying out. That's why a lot of times what do people do that have that type of situation? They put talcum powder. And what is that? It's a drying agent. That's all it is. It's absorbing the moisture, right, to help remove the moisture. Babies who get diaper rash, again, it's normal fungi, yeast that you have associated with your skin. The problem is we put a diaper on there, right? And we create a warm, moist environment if you don't change that diaper fast enough. Which, I'll tell you, as being a mom myself, sometimes it just can't be avoided, no matter how fast you change that diaper. Right? It's going to happen. And what do you put on it? Drying agents to help dry it out. Okay, so as we said, right, this competitive exclusion, right, is really important. And the ones that live in our skin that degrade the lipids, they actually produce fatty acids that are toxic to others. The E. coli, as I said, is going to compete. Lactobacillus is what helps keep the vagina really low pH and outcompete other bacteria. We've just established a really good relationship with them. Candida albicans is the yeast, though, that will create problems when the bacteria population is knocked down. And Clostridium difficile will create the problems in the intestines. A lot of times after, extreme antibiotic treatments. So toll-like receptors we've talked about. We haven't talked about complement yet. So let's look at complement system and what do we get? What's the benefit of this system? So I'm going to go ahead and play an animation for you guys. And again, I want you guys to focus on what happens, right, when this system is activated. What's the benefit of this system?
Regardless of whether the complement system is activated by the classical, alternative, or leptin pathway, the activated complement proteins are involved in several important processes associated with host defenses, including inflammation, attraction of neutrophils, opsonization, and cell lysis. Complement C3A participates in inflammation by inducing changes that cause the walls of local blood vessels to become more permeable. This facilitates movement of phagocytic cells from the circulation into the tissues. Complement 5A induces a directed chemotactic migration of phagocytes to the site of complement activation. Complement C3b functions as an opsonin. It binds to microbial cells, making them more susceptible to phagocytosis. Complement proteins C5b, C6, C7, C8, and C9 react together to form a hole in the cell membrane. When C5 convertase converts C5 into C5a and C5b, the C5b binds to the cell surface and combines with C6. C5b6 binds C7, enabling it to insert into the membrane. Binding of C8 is followed by binding of several C9s. The hole generated by formation of this membrane attack complex causes the cell to lyse.
So if we get the formation of this, the membrane attack complex, as I alluded to earlier, we can directly destroy pathogens with complement. So what is complement? It's a series of 9 proteins. All of them, of course, are denoted by the letter C to denote for complement. They're numbered in the order in which they were discovered, not necessarily the order in which they're activated, right? This is a cascade of events, right? So one thing starts, it's like a domino effect, right? And then other things down the line will happen. Some of the proteins break into 2 fragments. So notice they said C3A. That refers to that it is a fragment of C3, right? It's a piece. C3 breaks into two pieces, C3A and C3B. And as we saw, right, that fragment, C3A, was really important for the inflammatory response, right? Because it interacted with the endothelial cells to make them more permeable for those white blood cells to come in. Anybody know what white blood cells they were showing? They had a multi-lobe nucleus. Neutrophils, right? And we know, right, those are the main ones that are going to be called on scene because they're phagocytic. And that was the next thing we saw, right? We saw them opsonizing. We saw them being destroyed because they'd been opsonized. And what I say when I say opsonize, I say tagged for destruction, right? Because literally it bonded to the cells, right? And then we had the MAC complex. So these are our major outcomes, right? An inflammatory response, we're going to get more white blood cells to the area. We're going to maybe even get opsonization, that tagging for destruction. And we may even get cell lysis by the MAC complex forming. So for the inflammatory response, we saw the C3A, how that helped with the endothelial cells. And do you remember what the C5A, what did they say that was important for? No, C3A was helping the endothelial cells become more permeable so the neutrophils will come in. Well, once the neutrophil come in, how do they know where to go? The C5A is a chemoattractant for the neutrophils, right? They go right to that C5A. C3B is the opsonizer. And so how I remember this one is I remember B for binding, right? It binds to the surface of the pathogens. And because it binding is going to elicit destruction by phagocytic cells, it's called opsonization. Basically tagged for destruction, right? It's like putting an X on a tree so that the lumber guys know which trees you want cut, right? We're going around, C3B literally puts a big old X on that bacterial cell. And the phagocytic cells, guess what? Have receptors specifically for C3B, right? That protein that we produce in the complement system. So they can recognize, grab on, and phagocytize those pathogens. So it's really beneficial to us. So the next is the MAC complex. And the good news is I'm not going to make you guys memorize all the letters and numbers involved in this one. But the previous one, C3A and C5A and C3B, you got to know them. You got to know what they are, what they do. You got to know those three. The next we're just going to refer to as the membrane attack complex. You'll commonly see abbreviated MAC, right? Not to be confused with a Big Mac. But remember, M here means membrane. And so this is really important because it only inserts into membranes. So that helps us understand this next statement. It says that it's most effective on gram-negative cells. It's less effective on gram-positives. So what's this structural difference as it relates to membranes that makes the gram-negatives more susceptible? What do they have? When we think about gram-negatives, what do we think about? They have a thin layer peptidoglycan that's usually between two what? Membranes. And what do we call that last membrane that has the LPS in it? Just the outer membrane. It's not that complicated. It's just the outermost covering though, right? So it's an outer membrane. So those proteins can insert right into that outer membrane. So we can destroy gram-negatives with the MAC complex if they can insert into that outer membrane. Well, for gram-positives, what membrane do they have and where is it? They just have a cell membrane, right? Where is it? How do you get to it? You have the cell membrane, and what makes gram-positives gram-positive? Peptidoglycan. Where is that? That's on the outside of the cell, right? You have cell membrane and then thick layer peptidoglycan. So these proteins have to go through the cell wall to get to the membrane. So less likely to happen, right? Because it's just not as accessible, that membrane, right? And the fact that they only have one makes it less effective on our gram-positive invaders, but really effective against those gram-negatives, right? Because they've got an outer membrane, which is good news for us, y'all, because that outer membrane is just evil. That extra layer of protection really helps protect them a lot of times. Okay, so next time, right, that's the key, right? Opsonization with C3b, inflammation, C3a and C5a, and the formation of that MAC complex that will cause cell lysis. Next time, we're going to talk about how we actually activate that system, right? How do we get the ball rolling? Okay? All right. See you guys on Thursday.